I'm Stephanie Taylor, and this project to dissect the mechanism underlying fuzzy dysregulation in C9orf72-related ALS was conducted in Professor Kevin Talbot's lab under the supervision of Stephen Chen. The fuzzy gene codes for fuzzy planar cell polarity protein and is interesting because it is downstream of C9orf72 and is dysregulated in other neurodegenerative conditions. We found that fuzzy is downregulated at the RNA level in iPSC-derived cortical and spinal motor neurons from ALS patients carrying the C9orf72 mutation. We model C9orf72 ALS using neuroblastoma cells transfected with varying lengths of the GGGGCC repeat. We co-transfect with plasmids containing the fuzzy promoter linked to a luciferase reporter, which gives a readout of the fuzzy promoter activity. This enables us to demonstrate that the repeat expansion acts on the fuzzy promoter producing downregulation and to determine the sequence within the fuzzy promoter that is responsive to the repeat expansion. We narrowed down this responsive sequence to a 250 base pair region in the fuzzy promoter, highlighted in red. This is between -1332 and -1082, where +1 is the transcription start site. The degree of downregulation is expansion sensitive, with longer expansions producing greater downregulation. We investigated the role of both dipeptide repeat proteins and expanded hexanucleotide RNA in fuzzy downregulation. We used constructs that solely produced specific dipeptide repeat proteins, and in separate assays, constructs that express expanded toxic RNA only. The downregulation of fuzzy, mediated at the sequence in red, was reproduced in response to the toxic dipeptide repeat protein, polyproline arginine. It was also reproduced in response to expanded hexanucleotide RNA, implicating both poly-PR and toxic RNA foci in the mechanism for fuzzy downregulation. The diagram on the right indicates the constructs used. The RNA-only construct includes stop codons to prevent DPR production, and the DPR-only constructs are codon-optimized. Using the PROMO and JASPR databases, we identified transcription factors that could be involved. The responsive sequence in the fuzzy promoter contains binding sites for the transcription factors YY1 and CEBPB. Mutating the YY1 binding site abolished the repeat expansion-mediated fuzzy downregulation, implicating this transcription factor in the mechanism. This is reproduced with the RNA-only constructs. In conclusion, we have dissected the mechanism for downregulation of fuzzy resulting from the C9orf72 repeat expansion, which could be significant in the cellular pathology of C9orf72-related ALS. Thank you.